welcome back to the control group. Today, I want to start with what might be the most important question this channel will ever ask. What actually is clinical research? Because here's the thing, people talk about studies, trials, and the science all the time. It shows up in headlines, on social media, and debates about food, vaccines, supplements, medication, you name it. But very few people ever slow down and explain what clinical research actually is, how it works, who runs it, and what it can and cannot tell us. So today, I want to walk through this carefully. Not as an authority, not as a clinician, and definitely not as someone with all the answers, but as someone who spends a lot of time reading this literature, trying to understand it, and figuring out what it actually means for real people making real decisions. If you, like me, have ever felt confused by medical headlines, skeptical of bold health claims that you see on the internet somewhere, or frustrated by how often this science seems to contradict itself, this video is for you. Okay everyone, quick disclaimer before we get into it. I am not a physician and I am not a scientist. I'm not here to give any sort of advice or tell you what to do with your health. What I am is someone who reads clinical research closely, tries to understand what's actually being shown and where it's strong and where it's weak. And then I like to share whatever I find openly here. And probably most importantly, I want you to fact check everything you hear me say. If you see something wrong, if you see anything that doesn't line up with the literature or with your own experience, please tell me about it. That's kind of the whole point of this channel. It's how we learn. All right, with that out of the way, let's start talking about what clinical research really is. Okay, let's first start with why this topic feels so murky in the first place. When most people hear clinical research, they picture one of two extremes. Either it's something shadowy and corporate, something like big pharma, secret trials, profit-driven science, or even some shade of conspiracy. Or it's treated like gospel, where published equals proven, study equals truth, case closed, nothing more to learn. The reality, though, is much less dramatic and, in my opinion, much more interesting. If I were to try to define clinical research for you simply, it's the systematic study of health and disease in humans. That's it. It's the process we use to answer questions like, does this treatment work better than what we already have? Is it safe, not just in theory, but in real people? Who benefits and who doesn't? And who might get harmed? And importantly, compared to what? Since clinical research is never happening in a vacuum, it's always comparing something to something else. Whether that's placebo, standard of care, another drug, another behavior, or sometimes nothing at all. That comparison is where both the power and the limitations of research come from. And to understand that, we need to talk about how clinical studies actually come to life. Contrary to popular belief, clinical research doesn't start with humans. It usually starts with a question, often born from basic science, observations in clinical practice, or sometimes pure curiosity. Let's say researchers notice that a certain biological pathway seems to be involved in inflammation. That insight might come from lab experiments, it might come from animal models, or it might come from genetic studies. Before a single human is ever involved, though, there's typically preclinical research, lab work, cell studies, and animal studies, designed to answer a very basic question. Is this even plausible? And if it is it potentially dangerous? Most ideas actually never make it past this stage. And that's important because by the time something reaches humans, it has already passed through multiple filters, scientific, ethical, and regulatory. Once researchers believe something is worth testing in people, that's when clinical research formally begins. Easily one of the most misunderstood parts of clinical research is the protocol. Every clinical trial runs on a written document, sometimes hundreds of pages long, that lays out exactly what will happen. This blueprint of sorts details who can participate, who cannot participate, what will be measured, when it will be measured, what counts as success, what counts as harm, how safety will be monitored, and how data will be analyzed. This is not something researchers make up as they go along. In fact, once the trial starts, deviating from the protocol is considered a serious issue. Not flexibility, but a potential threat to the integrity of the data or patient safety. This rigidity is frustrating, and anyone who's worked in clinical research can tell you it can be an absolute pain sometimes. But it exists for a very good reason. It protects participants and it protects the validity of the results. It also explains why researchers get a little tense when someone says, "Can we just tweak this one thing real quick?" However, before any of this can happen, someone has to decide whether the study should happen at all. This is where institutional review boards, or IRBs if you've heard of the term, come in. An IRB is an independent committee whose sole job is to protect human participants. They don't care if the study is exciting, they don't care if it's expensive, they definitely don't care if it might change medicine. They care about risk, consent, and ethics. They ask questions like, are the risks reasonable compared to the potential benefits? Is the consent process honest and understandable? And finally, are vulnerable populations protected? No IRB approval, no study, period. And this is not a one-time check. IRBs continue to monitor studies, review adverse events, and can pause or stop a trial if concerns arise. Once a study is approved ethically, it moves into the phase most people are familiar with, the trial itself. Since you clicked on this video, I'm going to presume you've probably heard terms like phase 1, phase 2, phase 3, often used interchangeably in media. However, they are very, very different. Phase 1 trials are usually small. Their main goal is not effectiveness, it's safety. Researchers are asking questions like, how does the body handle this? What dose is tolerable? And what goes wrong early? Phase 2, things get a little bit more interesting. They start asking whether the intervention actually does what it's supposed to do, but it's still in relatively small groups. Phase 3 is probably what most people think of when they think of clinical trials. These trials are larger, longer, and designed to answer a much harder question. Does this meaningfully improve outcomes compared to existing care, or standard of care? Even after approval, phase 4 studies continue monitoring real-world safety and effectiveness. This structure exists because biology is messy, and early signals often do not hold up when tested rigorously. And that brings us to how results are actually interpreted and misunderstood. When a trial ends, researchers analyze the data according to the plan laid out before the study began. This is where terms like statistical significance, confidence intervals, and effect sizes come in. Topics we'll cover later on. But here's the key idea. A study does not deliver truth. It delivers evidence under specific conditions. A result can be statistically significant, but still clinically trivial. A result can fail to reach significance and still be informative. A result can be true in one population and misleading in another. This is why single studies should rarely change how we think overnight, and why replication is so important. And yet, this nuance is often lost once research leaves the journal. By the time research reaches the public, it's often been compressed into a headline that strips away most, if not all, context. Absolute risk becomes relative risk. Small effect sizes become breakthroughs. Early findings become absolute certainty. And to be fair, this isn't always malicious. Sometimes it's just the economics of attention. This is why understanding how clinical research works is so important. It's why I'm making the video. It gives you tools to pause, ask better questions, and resist overconfidence. So, what does all of this actually mean for you? Please, don't get me wrong. I don't think understanding clinical research makes you immune to misinformation, but it does make it harder to fool you. It helps you recognize uncertainty without panic, it helps you spot overclaims without becoming cynical, and it helps you stay curious instead of defensive. And honestly, I'm still learning this stuff myself. Every paper I read reminds me how much we don't know and how much I, as a person, absolutely do not know. But that humility isn't a weakness of science, it's the entire point. Thank you guys so much for spending this time with me. If this video helped clarify how clinical research actually works or raise new questions, I'd love to hear them. Comment them down below. If you disagree with something I said, please tell me. If you think I missed an important nuanced point, please, again, point it out. If there's anything, a new study, a new paper, a new methodology you want to walk through together, drop it in the comments and I'll see if I can make a video about it. This channel isn't about having the final word. It's about learning in public together. I'll see you guys in the next one.